The peptide tag should be removed too, because they can interfere with the protein activity and structure. This we understand. If with your protein of desire, some other tags are there, peptide tags or fusion partners are there, they may interfere with their activity or their actual structure. But they can be left in place even for crystallographic study. In some of the stances, they can be left as such. Tags can be eliminated by either enzymatic cleavage or chemical cleavage. We have both the options. We can use chemicals as well as we can use some enzymes. In the series of enzymes, the examples are like enterokinase enzyme, thrombin, coagulation factor 9A, and the tobacco H virus proteases have all been successfully used for the removal of peptide tags and fusion partners from the proteins. Choosing among the different proteases is based on specificity, cost, number of amino acid left in the protein, and all these points we had already talked in the previous class. That how you finally settle down with a particular enzyme that you want to use. Definitely, cost is very important, as we will see in the coming slides. That thrombin is very costly. Why? Because recombinant productions are not possible yet now. Only sources from the plasma of the animals or the bovine. So it is very costly. Other things are there, that how once you have used the enzyme to remove a tag, you should get rid of this enzyme also from the sample. You cannot leave the enzyme there itself along with your protein. So there should be some mechanism with which you can remove this protein after its action. And another factor, that is number of amino acid left in the protein. So for example, if suppose after the cleavage your peptide has been removed and fusion partner has been removed, then usually we see that these enzymes, they act on not one single amino but rather a specific sequence of amino acids. 2 3 amino acids may be there. So if it is digested, if it is cut, it may leave there a few amino acids. So naturally you would like that after the digestion, after the removal of the tag, uh, not many large number of amino acids should be left along with your protein. That will again interfere with the function of your protein. So for that reason, we want that minimum number of amino acids should be left, if at all, after the cleavage by the particular enzyme. Enterokinase and thrombin were popular in the past, but the use of his tagged TEV has become an everyday choice due to its high specificity. It is easy to produce in large quantities and leaves only a serine or glycine residue or even the natural N terminus sometimes after the digestion. So it says what are the relative advantages of using TEV in comparison to enterokinase or thrombin. About enterokinase. What is this? Enterokinase enzyme, as we all know, it cleaves its natural substrate, that is trypsinogen, to form trypsin. In our intestine, stomach, trypsinogen is there, and it is a pre-protein kind of structure, trypsinogen. So to make active trypsin from this, it needs to be cleaved by the enzyme, and that enzyme is enterokinase. In some of the literature, you will also find that it is called enteropeptidase or enteroproteases or enterokinase. Enterokinase is highly specific mammalian serine proteases that recognizes the amino acid sequence, that is aspartate, 4 aspartate residues followed by lysine, and I think this arrow has shifted. Arrow should come here just before X. So this is cleaved. Wherever this sequence will be there, it will be cleaved here immediately after lysine and before any amino acids. So if our fusion partner and our protein of interest protein of interest in between them, if this site is there, it will be recognized by enterokinase and it can be cleaved. It is also said that although this is a consensus signature of the cleavage by enterokinase, the partial substitution of aspartate residues by It is by mistake. It is GLY, glycine. By glycine is possible. That is the cleavage site. Glycine 4 can also be cleaved at this position, is also recognized. So a minor change from aspartic acid to glycine can happen. That will also be recognized by the glycine. And when we say that this type of sequence is recognized by enterokinase and that cleaves our protein and fusion tag, these type of sequences are usually rare in our gene of interest. Naturally, then this enzyme will not cut your protein. It will not cut our protein, but rather it will cut at a specific site where it has been added or introduced. So it says these sequences are relatively uncommon in the natural proteins. So whatever proteins you are trying to express, they do not have this site. Thrombin. Thrombin is typically purified from bovine plasma. See, the source is bovine plasma. Although recombinant human thrombin has been produced in Chinese hemisphere already. So what it says that we have been relatively successful in expressing this thrombin enzyme in the CHO cell line, Chinese hemisphere ovary. So it is a mammalian expression system. And now you know that if you want to produce any enzyme or protein in large amounts so that it can be used commercially, usually you prefer bacterial host because the amount is not limited. You can generate lot of amount of that protein. But here, some of the researchers, they have tried to express thrombin in CHO, that is Chinese hemisphere. So naturally, although it is expressed in Chinese hemisphere ovary cell lines, but the yield is not sufficient. Yield will not be sufficient for, and the use was for certain clinical applications where it is more demanded, and there people can, in the clinical applications, people can afford the higher cost. But expression itself in the expression mammalian system itself is very costly. As of now, no convenient procedure for expression and purification of recombinant thrombin is available, and therefore it is highly costly. The thrombin enzymes, they usually prefer this site, the consensus sequence which should be present between your protein and the fusion partner or peptide tag. It should be leucine, valine, uh, proline, arginine, this is glycine, and then follow serine. So before glycine and after arginine, it will recognize the site and it will digest. Thrombin can be inactivated. So as I said in the previous slide also, that once we choose an enzyme, we should also have a mechanism with which you can remove those enzymes which you have used for cleaving your product, cleaving your fusion partner. So either you should have some mechanism to remove it by purification, by chromatographic methods, or you should have certain mechanism with where you can, if you add something, it should be inactivated. It should not be able to act further. So thrombin can be inactivated by PMSF. We know PMSF is used as a protease inhibitor, and it is also used to keep your protein sample safe for longer period of time. If you have extracted protein from any sample, and if you want to store it at room temperature or 4 degrees Celsius or minus 80 degrees Celsius, then you would like to add, usually you add PMSF in that, so that all the proteases present in your sample, they are inactivated and your protein therefore will remain safe. So PMSF is added, which inactivates this thrombin, or it can also be inactivated by ABSF, benzyl sulfonyl fluoride hydrochloride. So this is another compound. So these two compounds can be used to inactivate the thrombin, which have been used for cleaving the fusion part. Other systems are also there where you can either remove the enzyme. Instead of inactivating in the sample itself, you can remove it. So for this, a biotinylated form of thrombin has been produced that can be absorbed on the avidin or streptavidin resin, is commercially available. So people have also uh, produced thrombin molecule which are already tagged with biotin. So biotinylated thrombin people are able to produce, and they are selling it commercially also. So when your thrombin is already tagged with biotin, so you can easily remove them by capturing on avidin columns or streptavidin columns. As you know, biotin has very strong affinity to avidin and streptavidin. So this biotin-related thrombin can be captured in columns. Commercially, columns are available. They are either avidin columns or streptavidin columns, and you can remove it easily. Two, three other points are there about thrombin. Although I have shared this paper, uh, it is a review article. You can read in detail about all these enzymes. They have been discussed in more detail than what I am showing you here on the slide. So the optimum temperature for thrombin activity is around 45 degrees Celsius, and the enzyme maintains 20% of its maximal activity at even 15 degrees Celsius. So there is a range. You can use from 15 degree to 45 degree for the cleavage of your fusion partner. The pH range at which thrombin is active, it is in the range of 5 to 10. And normally with the optimum pH of about 9.5 in the absence of NaCl and 8.3 in the presence of 1 molar NaCl. So these are certain chemical condition at which these enzymes are. Now, another important point is that as it has disulfide bond, so can be inactive in the presence of reducing agents in the medium. So what happens usually sometimes when you have purified your protein by following a series of chromatographic methods, then you want to cleave off the fusion partner. You should always remember that if you are using thrombin enzyme, since thrombin enzyme has disulfide bonds for its proper activity, for its proper 3D structure, there are disulfide bonds present in them. So if these disulfide bonds are removed, are broken, then it will become linear. And the linear thrombin, then linear primary structure of this thrombin protein will not no more be active. So it will not cleave off the product. It will not cleave off the product. So that is why it is said that the reducing environment should be avoided. If in your the protein in which you are keeping your sample, you should be careful that DTT or some other thiol type of thing, they should not be there, which are reducing in nature. So if they are reducing compounds, what will they? They will break the disulfide bond. So these are two, three chemical characteristics, chemical conditions. What should be the temperature? What should be the pH? And what should be the chemical environment? It should be kept in mind. Another enzyme that you read in the previous slide, that tobacco itch virus TEV proteases. So actually this TEV protease is an inclusion proteins which are found in this tobacco itch virus. And they are also called as nuclear inclusion protein, termed as NIA. Nuclear inclusion, A is a subscript to designate that protein. The protein's activity resides in its C-terminal 27, although it's a bigger protein. It's a big protein, if I remember correctly, around 45 kilodalton. But the actual protease activity in this protein lies in this 27 kilodalton domain, and therefore people have been able to produce only this much fragment, 27 kilodalton, and this can be commercially used. If you look at the sequence at which it digests the protein, it is ENLYFQ followed by glycine. So between this glutamine and glycine, the cleavage site is here. It recognizes human rhinovirus 3C proteases. Now see, this is another enzyme which is very often used. And as the name suggests, human rhinovirus, it is a very common virus. In fact, it is mostly involved in all types of cold, common cold, which people, human beings, they suffer. From those viruses, in fact, if you look at the family of these viruses, they belong, they share even poliovirus, this common cold virus, SARS virus, they all belong to the same group. And therefore, people have no answer that why how this virus only became popular. The
Sir, we cannot hear you. Hello?
Sir,
You are on
Yes,
mute.
yes,
Please
I will be
unmute.
back. I will be back. हाँ sir. Yeah, sorry, I had a call. So, this is about human rhinovirus 3C proteases. Sometimes you will also find in some of the research papers that human rhinovirus 14C proteases are there. These are 14.3 and these are the strains of rhinoviruses. Here also 14.3C proteases is commercially available in the form of glutathione as transferase fusion protein. Again, you see here, these proteins are commercially available as a fusion partner again, because with them, ultimately at the end, you would like to remove these proteins from the sample. So how you can remove? Because they have this glutathione, GST. So you can use any reducing substances, glutathione, and it will be captured by the glutathione. And then it can be removed from the mixture. and and the commercial name, this is a commercially available product which are this 3C proteases and tagged with GST. The commercial name is precision proteases, and it is very popular in many of the laboratories who are working with the recombinant protein production. They use they call precision proteases. So as such, name does not indicate what is precision. So precision is actually about human rhinovirus 3 proteases, and they have all these viral proteases, as we saw TEV proteases and these human rhinovirus 3 proteases, they are very specific in comparison to the other proteases which saw like thrombin or coagulation factor 9A, like that. So they are better than them. And the recognition site is usually ETLFQ followed by GNP, glycine protein. But in some of the literatures, there are many other sites also they have been reported with, of course, a little bit of less or more efficiency. So therefore, this site is also reported so far so in the documentation literature of Precision is concerned. So Precision's the commercial company, the vendors who sell it, in their literature you will find this sequence, but otherwise in the research paper you will find this sequence is recognized by not much difference, but a minor difference. But I think both of them, they are recognized pretty well by this 3C proteases. And this slide, this photograph you had already seen that how in such some of the vectors, these sites have been already incorporated to remove the tags, for example, from your protein, enterokinase site, HRV3 sites. Similarly, here enterokinase site, here HRV3 is a thrombin site. So many uh, ways are to show that these vectors, they contain the enzyme amino acid residues which are recognized by the particular cleaving enzymes. Removal of the tags by chemicals. I think in the last class we had also talked about this, that the most common chemical is cyanogen bromide. So cyanogen bromide can be used, but still I would like to go through the advantage of using chemicals for this purpose. Is to eliminate from the reaction mixtures. They are cheap in comparison with the proteolytic enzymes. Chemicals are very cheaper in comparison to enzymes. However, the reaction conditions are very harsh if you are using chemicals. So their use is largely restricted to purify recombinant proteins obtained from inclusion bodies. Why inclusion bodies? Because usually we see that inclusion bodies amount in the bacterial system of expression, the amount in the form of inclusion bodies are quite high. So if you have much amount of protein, you will not bother about loss of your protein. So you can use chemical based method, which is harsh. Even if some of the proteins uh, they are made non-functional because of the harsh treatment of the chemical, still you will have sufficient amount of proteins for your use. They also often cause unwanted protein modifications, these chemicals. The most common chemical cleavage reagent is cyanogen bromide, and it cleaves the bond C terminal to the methionine residue. So this is important about the action of cyanogen bromide. It cleaves C terminal of the methionine. For example, if you have a protein followed by fusion tag. So if your protein, mind it, if your protein at the C terminal ends with methionine, then you can expect that methionine, just immediately after methionine it will be cut. And if it is not a, nowhere in your protein, then you can add any 2 3 more residues and you can add methionine in there. So after methionine it will cut. So this amino acid should be present between the tag and the protein of interest. Also, the target protein should not contain internal methionines naturally. It should not. Cyanogen bromide cleavage can be performed in common denaturing conditions. So these are the chemical environment at which it can be performed. So 6 molar guanidine hydrochloride or 70% of formic acid or trifluoric acid is should be there. Other chemical methods for protein cleavage can be this. I will suggest that you read this Huang et al. 2014, or otherwise I have given you, or otherwise I have also given you a review article that you read. To perform this CNBR cleavage of fusion protein, the majority of the researchers first desalted. So usually when you want to go for cleavage with cyanogen bromide, initially in the past people have been using purification of their product. Purification means all the salts were removed and then vacuum dried the samples, and which were then in turn they were dissolved into a proper aqueous solution which contains formic acid and trifluoroacid. But later on people came with the report that say that this desalting step is not essential step. You can use cyanogen bromide based removal of the cleavage, removal of the fusion partners, even in the presence of salt. You need not to clarify that. However, desalting step can be omitted and run cyanogen cleavage directly. So it can be done. The commonly used tris HCl, sodium phosphate, NaCl, imidazole, and guanidine hydrochloride do not interfere with the reaction under acidic condition. So these are some facts about the mechanism of action of the cyanogen bromide and the chemical environment it is where it is able to function. So now this is about host cells. Last slide, last class, I think we had left here. This is last 2 slides. I will finish today. For a first expression screen, only a couple of E. coli strains are necessary. That is BL21 and some derivatives of K12. So these are the very commonly used E. coli host cells which are used in lab. One is K12 lineages cells and other is BL21DE3. But both of them are E. coli. Remember it, both of them are E. coli cells. The history of the BL21 and BL21DE3 cells has been documented in this. If you want to read about more in more detail, although I have shared with you one review article about the E. coli host modifications. How over the time in the centuries of years, how lot of genetic mutations have been brought down in these host cells to make it best and more suitable for expression of your protein in those hosts. So a lot of mutations are carried out, in fact. So we will read a little bit of one or few salient points. So BL21 cells are deficient in the lone protease. So lone protease is an important enzyme which degrades many of the foreign proteins. So bacteria usually have their own system, own mechanism. So that is for their protection. So whenever any foreign proteins in the form of phages or bacteriophages they come, then bacteria employ those as a weapon. So these, if these weapons are there in your host, then when you are trying to express your protein, then also it will degrade your protein and you will not get. So what you need, that you desire that such proteases should be absent in your host cells in which you are trying to express your protein. So lone protease is deficient in BL21. Another gene missing from the genome of the ancestor, another protein which is missing from the genome of the ancestor of the BL21, ancestors of the BL21 from which BL21 has been made, is the one coding for the outer membrane protease OMPT. Again, this is another protease, OMPT, outer membrane protease. So this protein is also mutated, is also be missing, whose function is to degrade extracellular proteins. The liberated amino acids are then taken up by the cell. So this is a natural mechanism, natural system in all the E. coli. So those E. coli are selected, mutated, selected in such a way that these proteases are missing from those host cells and they can be utilized for expression of our gene. This is problematic in the expression of recombinant protein, as after cell lysis OMPT may digest our protein. Very clear. In addition, plasmid loss is prevented because, again, there are certain hosts, E. coli, which were able to uh, degrade your DNA. DNA will be chopped off. So if my plasmid DNA is chopped off, then how can I expect expression of protein from my plasmid? So there, and you know that almost all the bacteria, E. coli, they have a mechanism of restriction digestion. So any foreign DNA will come, it will chop off. So to avoid this chopping off of our DNA, of our plasmid DNA, there should be mutation in certain genes, and that mutation is in HSDSB mutation. So this is a very important mutation to be present in the host cell. Uh, so this is already present in the parental cell line, that is B834, that gave rise to BL21. So BL21 which is coming from this B834, and in that strain there was already a mutation in the HSDSB. So because of this mutation, it will not be able to degrade your DNA. It is mutated version. Its mutated version prevents cleavage of the external DNA while still protecting its own DNA by methylation. So although this mutation prevents the degradation or the enzymatic cleavage of the plasmid DNA, but it will protect its own DNA from the cleavage because it is still able to carry out methylation. And you know that if it is able to do methylation, then it will not be cleaved by those restriction injects. When the gene of interest is placed under T7 promoter, and now again back to this, as I had told you in the PET series of vector where you say T7 promoters are there. So what I had told, T7 promoters are activated by RNA polymerase. So where from RNA polymerase will come? RNA polymerase usually comes from the genome, in the host genome. So they have been either integrated into the host genome or they have supplied in the form of vectors. So in this case, there are certain E. coli hosts where it says that you have already integrated T7 RNA polymerase under the lac UV promoter. So it says that usually in the popular BL21 or DE3 strain, the lambda DE3 profiles was inserted in the chromosome of BL2. So now you need not to supply it from outside. The RNA polymerase under the lac UV5 you are supplying from outside so that it can be induced by lactose or IPTZ. So you do not require this. So it is already present in the chromosome of the host. So whenever you are putting your vector, your plasmid DNA into this, and you will put you will add inducer IPTZ, automatically RNA polymerase will be transcribed, and this RNA polymerase will then go and it will bind on your plasmid DNA, on your recombinant plasmid DNA at the T7 promoter, and then your gene will be transcribed. So there are these changes in the host cells which are already made, and these are genetically engineered host cells, and they can be well utilized for the expression of the protein. Still, there are reports where the K12 lineage is used for the purpose. So this was about BL22. Now K12 lineages are also available, and the typical examples in this series are the AD494 and Origami from the Novaji. So Origami strains are also sold from this company. They are from the K12 strains, K12 lineage. These are thioredoxin reductase mutants. So disulfide bond formation in the cytoplasm is enhanced. So origami, if somebody asks you why you prefer origami strain, because they have already mutated thioredoxin reductase. So what happens if thioredoxin reductase is mutated, it will not be able to cleave the disulfide bond. So if your protein, suppose it is insulin, if your protein contains disulfide bond, then it will not be able to reduce it. It will not be able to cleave it. So this mutation is important, and for that purpose, origami strains are useful. Also lacks the glutathione reductase enzyme. So there are two enzymes it lacks. Another widely used strain from the K12 repertoire is HMS174, a RAC mutant. Again, this is RAC mutant. There is recombination. This mutation has a positive effect on plasmid stability. So recombination should not happen. So for that purpose, also there are certain mutations. Another one is plasmid multimers formation is an important cause of instability. Okay? हम लोग कभी देखते हैं कि plasmid loss हो जाता है, इसको कहते हैं plasmid curing. So plasmid curing क्यों होता है? Because of the formation of the multimers. वो concatenaric plasmids बन जाते हैं, one circle, another circle. So many circles of the plasmids they get like multimeric. So multimeric formation will be responsible for the loss of your plasmid from the cells. So how you can get rid of this? Again, doing certain mutations in that. So plasmid multimorphism relies on the recombination so system of the E. coli. So if you are using this mutation HMS174, it will prevent that instability. All the three strains have their lambda DE3 containing derivative so that T7 RNA polymerase can be used. So again, in origami strains, in these origami strains, you see that T7 RNA polymerase has already been put into the chromosome of the DNA. Now this is last slide, I think. So choosing a perfect combination. Now class is also finishing. Do we have time? Next 1 hour?